And I think this pioneering work that was done at the time, what's going to happen in 15 or 20 years with those patients that receive those cell therapy, an extremely important component of what cell therapy is today. Many people were saying this is never going to work. And we believe with my co-founders that it has actually a very important role, particularly in some immunosuppressive aspects that the tumors are generating. Engineering some of them, we can eventually really improve the outcome for cancer patients. If the immune system has created all those immune cells, there is a reason.
Welcome to Industry Voice series. In this episode, we're going to cover one of the most in-demand fields of clinical research, cell and gene therapy. Our guest today represents a cutting-edge cell therapy biotech, which is developing a novel approach to directly target leukemias. Please meet the CEO and co-founder of NKILT Therapeutics, Raphaël Ognar.
Raphael, thank you very much for joining us today. We appreciate it and are very much looking forward to our conversation.
Thank you for having me. I'm very pleased to join you and happy to discuss about my experience in the industry and in cell therapy.
So, Raphael, you have a very outstanding career working for various big pharmas and other interesting companies. But today I wanted to talk about how you ended up in the field of cell and gene therapy. Could you please elaborate on that a little bit?
Absolutely. I've been in the industry for now almost 25 years. Started my journey in oncology around 2003 when I joined Novartis Oncology. And started in France to launch a new indication of one of their blockbuster drugs, and then very rapidly moved to the global headquarters in New Jersey to be able to expand my role to a global level, and rapidly pivoted to new product strategy roles where I was in charge of multiple assets in the pipeline from phase 1 to phase 3. This gave me the privilege to lead several programs, not only across multiple tumors in cancer, but also understanding, all the dynamic and all the complexity of drug development and what was needed to be achieved to bring a drug to approval, which I had the opportunity to lead 2 programs that went to approval in multiple myeloma and non-small cell lung cancer. So at that same period, there were some growing work happening in the background between Novartis and UPenn, with the lab of Carl June. And this is kind of, if you fast forward a few years from there, this is where the first CAR T therapy came out, leading to the first CAR T being approved. And there was a striking story that was really something that for some of us that were kind of course, doing our job in the new product strategy group, but always keeping an eye on this very interesting approach that the Carl June's lab was doing. And when came the first treatment of Emily, that young 6-year-old girl that was suffering with ALL, everybody was on the edge following every single week about what was happening to Emily. We know that it was not a smooth ride in the beginning because it was the first time a CAR T cell therapy was really given to patients. And after several weeks of ups and downs, she started to see some responses. She started to be feeling better. And we just celebrated actually this year, the 10-year anniversary of her first treatment. And she's a beautiful 16-year-old teenager now that is very active in the field. So at that time, there was really something that got kind of like seeded in my mind about cell therapy. And I started to look upon it with more consistent manner. Of course, what was really great is that I had the opportunity to see that and witness cell therapy even closer on my next move when I moved out of Novartis and I went to Celgene to work in corporate strategy. At the time, Celgene was starting to engage with Juno, that is now kind of the cell therapy arm of BMS that has acquired Celgene in the meantime. So I kind of had the opportunity to really analyze that market. I actually spent 6 months of my time doing a deep dive of immuno-oncology and cell therapy at that time to provide a report to the C-suite of Celgene at the time. And that really allowed me to force myself to understand what was happening, but also being exposed to the incredible science that was behind and the promise that we were trying to achieve with this. So fast forwarding, I ended up moving out of corporate pharma and biotech, and I created my own consulting. And through that consulting, I focused essentially on early stage companies, trying to help them moving to the next level. And throughout that journey of almost 5 years, I met a lot of people, great people, but I also had the opportunity to work in the cell therapy field, especially a couple of projects, of which one I was an acting CEO, so consultant CEO, to build the foundation of a cell therapy company. And I met a ton of great people in that specific field that just reinforced my interest and allowed me to continue to learn. And through these multiple meetings with the great people, I actually met my two co-founders, or the one that became my two co-founders for our cell therapy startup, NKILT Therapeutics, that we founded last year in summer. So this is really how I kind of like, it's very, very high level summary, but how I landed there. I just want to point one very important factor is that through that journey, I had the privilege of having someone by my side that was very strong, not only personal partner, but also someone that is in the industry. That is Deborah Barton. She's a medical oncologist. She's a very highly experienced clinician, and she's also working in the field of cell therapy. She's actually Chief Medical Officer currently at TScan, and she was part of the team that also pushed a very new approach in cell therapy when she was working for Charisma, where they pushed out the first CAR macrophages in the clinic, coming also from the lab of Carl June, and Charisma was co-founded by Mike Krzyzewski, that is one of the creator of this technology. So thanks to her extraordinary knowledge and the fact that she was always supporting me when I was trying new things, particularly in cell therapy, this is really one additional key factor that made me where I am today. And I'm very grateful of that.
And that is a very impressive journey, I have to admit. And since you have a very vast experience in the field, in your opinion, what are the key developments in the history of cell and gene therapy products for cancer treatment and prevention?
So it's really that's a tricky question, because there's so many things that happen, and I'm sure that whatever I'm going to highlight, I'm going to miss some of the key milestones. Some people would say, "Oh, you should have spoken about this or this." So I will full disclaimer, this is a biased view. It's my view, and I'm sure I'm going to miss some things. But based on my story, what I could say is that I will always have to go back to Rosenberg and his teams back, a long time ago, when they were starting to do a lot of work on cell therapy, on TILs. And I think this pioneering work that was done at the time, where I always remember a lecture he did at AACR last year, where he highlighted some of his notes that he was taking at the time, and where he was kind of literally hammered by FDA at the time, saying, "You are trying to play God, you are touching things you shouldn't be touching," whatever. And in some way, I can understand the fear of what we were doing, engineering immune cells or things like this. People were kind of afraid, and that's totally understandable. But the preliminary work that he did there, and that he continues doing, by the way, today, I think has been an extremely important component of what cell therapy is today. Of course, along the way, many other great milestones happened. I mentioned Carl June Lab and the first CAR T approved in ALL. Obviously, this was a very strong moment. I remember at the time, many people were saying, "This is never going to work. That's great research, but it's never going to be able to go to patients, too complicated." And it's true, it is very complicated, but I think it was a kind of a very important moment where, for the first time, we had an approval of a cell therapy And now we have this commercialized thanks to Novartis that has taken over and that has put all the logistic and the support behind it. If I move forward, I think there was as I mentioned, one of the difficulty we face in cell therapy, particularly in the autologous approaches, is that the manufacturing processes are extremely complex and tedious and very costly. Of course, it's really the pinnacle of medicine, and specifically of precision medicine, but it makes it extremely complicated to manufacture. And there are many situations where it's unfortunately not necessarily the best option for some patients. I know we're going to talk about this later on. So I think what came as another really important milestone is when people started to think about moving out of autologous, which is a good option that would continue to move forward, but starting to look at the allogenic approach in order to try to create an off-the-shelf cell therapy concept. And that started to see other cells than the T cells being used, particularly the NK cells that have been identified as much better to address the allogenic approach and challenges. And from now on, we have so many companies that have embraced the NK cells and are trying to develop cell therapy with those. And that really opened up to the idea that why just using T cells or NK cells? Why not looking at other immune cells and see how they can contribute in some way, which I think is great, because if the immune system has created all those immune cells, there is a reason. They're all needed. They all have a specific role. So we should be looking at each of them and see how by engineering some of them, we can eventually really improve the outcome for cancer patients. And I'm not going to go back to the charisma story with CAR macrophages that I already mentioned, but that's another type of immune cells that are now being developed and studied. And so all this, to me, in a very rapid and holistic way, are some of the milestones we can see. And I'm sure there's so many more on the science aspect. But I think if you go back 20 years, I don't think many people would have put a dollar on cell therapy as a therapeutics. As a research, for sure. As a therapeutics, I think there were a lot of doubts. And I think today we are proving that it is something that we will make evolve and become a strong component of treating cancer.
And well, actually, if we stay on this topic, how do you envision gene therapy technology evolving in the future? Since you already talked about the past, now let's talk a little more about the future, maybe like over the next 5 to 10 years, for example.
Well, so that's actually a very, very tricky question. I would call that the billion-dollar question. But I think we're just seeing the tip of the iceberg currently about cell and gene therapy. And again, like I just said, if you look back at 10 to 15 years ago, I don't think we would be any close to anticipating how fast and how really how many doors we have opened and how much of understanding we have taken from our different studies that we did in the past years. What I found striking with the work we're doing in cell and gene therapy today is that it's almost every time we answer a question, we reopen a door with 20 more other questions, which is sometimes scary, but it's also fantastic because it means that we are pushing the boundaries and we're getting deeper and deeper in the understanding of what's happening in the immune system and in the human body. So if I were to fast forward and do some science fiction, I really believe that the addition of technology in general, the fact that we if you think about nanotechnologies, for example, that could help bringing more specific therapeutics in very specific places in the human body. Or if you think about just the engineering of immune cells and the capability we have to now control, create switchers that can, switch on, switch off the cells, all these things that probably many years ago were considered completely crazy, I think are going to become routine, and we will know more about it. and we will be allowed to really continue to develop new approaches with cell and gene editing to tackle cancer. I'd like to add one more specific aspect also, is that I think we are also growing beyond just the therapeutic aspect. We are more and more considering that innovation is not just about bringing a new therapy. Innovation is about being better at prevention. You asked a question earlier about prevention. I didn't mention a lot about this, but there are a lot of things we can do in anticipating or limiting the risk by better knowing what are the risk factors of different cancers, for example. This is part of innovation for me. Another big part of innovation is obviously diagnosis. The earlier we can diagnose, the more chances to cure cancer we have. We know that's a kind of a classic paradigm, but it has been proven that if you can stop it earlier, you have more chances of getting rid of it. And of course, then there's the treatment aspect, which I believe cell therapy and gene therapies are going to be a component whatsoever. And then a last point that I would not want to forget, that I also consider part of the innovation, is monitoring and engagement. I think we have understood that we needed to move away from doing healthcare for patient, from doing healthcare with patient. And making the patient a strong actor and very engaged is going to increase dramatically the outcome of what we are doing. And it's going to allow our industry to be even better at what we're doing. So I would say, for me, I would see the future of cell and gene therapy as the future of healthcare by really hammering this definition of innovation that I have, that is prevention, diagnosis, treatment, monitoring, and engagement.
Thank you for such an interesting point of view. So probably in the near future, we will see how it turns out. You already started talking about immunotherapy, and I wanted to go back to that question because I think that it's very important to talk about it. So is it the right treatment for everyone?
Yeah, that's actually a good and tricky question also. I think first I'm going to focus on cancer because that's where I have most of my experience. Immunotherapy has been also shown to be efficient in many other therapeutic areas. And I think this is also just the beginning, just to be very clear. And we are learning a lot of things in cancer that I'm sure are going to be applicable in other TAs. What I would say, is it the right treatment for everyone? Probably not as of today. There are probably situations where there will be other options that might be more appropriate based on the current state of science and knowledge. If I take a specific example, for example, in cancer, we do see sometimes issues with advanced cancer where, if you think about the autologous approaches in cell therapy, where you need to harvest the cells from the patient, then you need to go through a tedious and pretty complex manufacturing process to engineer the cells and then bring them back. It makes what we call the vein-to-vein time sometime pretty long, of a couple of weeks or 3-4 weeks. That could be a significant issue when you have a patient with advanced or bulky disease that will maybe make them no longer eligible or able to receive therapy because they would have progressed too much, or they will be too weak, or there are many other reasons. So that points out to the fact that there are situations where it may not be the good choice. We might still need, and I think we still need to use chemotherapy, for example, or targeted therapy. Many people believe that cell and gene therapy are not good for bulky disease, and that you will still need to use chemo to debulk and reduce the burden. All this is true, and I never believed in the one size fits all. So I think, will cell and gene therapy be part of the treatment strategy? Absolutely. Will it be the only option? Absolutely not. I think we will continue to have to play with multiple approaches between the combination strategies or the sequencing strategies, or both together. But I think it will be used, but I don't think as of today it is to be considered the right thing for everybody. There are also a lot of things that we have yet been able to understand is we are touching the immune system. What's going to happen in 15 or 20 years with those patients that receive those cell therapy, for example, or gene therapy? We don't know yet. So that's why FDA is really very strong in asking all our companies developing cell therapy to continue to monitor patients beyond their initial treatment, because we have to be sure that the intervention that is happening early on in the immune system is not going to trigger other things that might be problematic. So I think it's what I find fascinating, and it's also the proof that we still have more work to do. And I'm convinced we will be able to use cell and gene therapy across multiple patients, but there might be situations where it won't be the first choice.
Thank you very much for such a thorough answer. I think our audience will learn a lot from this. And of course, from a CRO perspective, I cannot help asking you a little more about the clinical trials. Are there any challenges in conducting cell and gene therapy clinical trials, and how do you overcome such challenges?
Well, yes, you do know very well, being a CRO, how difficult it is already to just put together a clinical program, regardless of the technology that we are using. Whether it's a small molecule, an antibody, or cell and gene therapy, it is by definition something complex and difficult, and it requires skills, talent, and a lot of rigor. In cell therapy, I think, particularly with the autologous approaches today, the bar is much higher, because you have this manufacturing, piece in the middle of it that makes it particularly complex, and it limits probably also the number of centers where you can do it. You need to bring the cells of the patient into a special manufacturing place. So these manufacturing facilities are not, everywhere in the world. So you can just imagine, if you have a patient in Asia and the manufacturing place is in the US, that's already by definition just a very big logistical burden for the clinical trial. So of course, the logistic is something that's going to be always a difficult aspect. And that's also probably why the idea of going with allogenic technology and off-the-shelf approaches is going to be one of the future. I used to say that the winners in cell therapy are going to be the ones that are going to be able to convert the incredible efficacy that we see in some autologous approaches as an off-the-shelf. I'm not sure we'll be able to do that, but that's going to simplify a little bit. Now, the other thing that I think that would be too simple to just say it's logistic and that's it. I think there are other challenges that we already see in classic clinical trials, patient recruitment issues, patient engagements, lack of diversity. We know that this is something that is already pretty difficult in a very classic approach for clinical trials. I guess it's probably emphasized in cell and gene therapy because those patients are even more difficult to find. And so it's going to require to have an increasing number of experts and a growing amount of expertise in our colleagues from CROs to be able to really master the complexity of cell and gene therapy clinical trials, and really become better at supporting the overall process. One thing also that is hopefully a short-term issue, currently there's so many clinical trials happening in cell and gene therapy that one of the biggest issues that we probably don't have enough manufacturing slot to be able to satisfy everybody in a diligent way. And that's going to be a potential bottleneck, that too much demand, not enough options to be able to have everybody doing everything the way they want. And there are more and more of the companies that are creating their own manufacturing suites because it's a better way of controlling it, but that's a humongous investment in capital. And so there's a trade-off that needs to happen. If you have to invest several millions into building a facility, those millions there are not going into other aspects of developing the science. So this is kind of a constant equation that we're going to have to solve. So I think improving our manufacturing process overall, improving the overall process development, maybe going off the shelf and allogenic, all these are going to make it easier. But I still believe that it will remain difficult to run clinical trials no matter what. And like I said in the beginning, we need to be better in recruiting more patients, in having more diversity in trials that are more representative of what the population is. And I think we're still far from that.
And what about the regulatory concerns regarding such trials and their analysis in treating oncology patients?
Well, I'd say the regulatory aspect is something that is really close to my heart, because I think it is, how can I say that? It's a very tough moment for our regulators in general. When you see the pace of innovation that our industry is bringing, and the amount of new approaches that we are bringing to the table, new immune cells, new transduction systems, there are a ton of new approaches that are coming in, and I am not sure that the regulators are able to keep up. First, it is obvious that if you take FDA, for example, they're completely understaffed. And for them to be able to have the same level of understanding of our technology that we are creating, it's extremely difficult. They need time, they need people, which they don't have today. And I really think that they're doing everything they can, but you can just see how it is difficult today to try to have an interact meeting that is a crucial first meeting to discuss with FDA about our technology, and how most of them are not happening because they just can't. They just don't have the resource to do it. And I pointed to FDA, but I think it's going to be the same in every other regulatory department across the world. And that's going to be a real problem, because we need a partner. We as industry, we need a partner as the regulator to really work together and say, like, we're bringing these new things. Of course, we believe in our technology, but we need to be challenged. But we need to be challenged by people that are, able to challenge us at the same level of what we are, and have a true sparring partner that can really be by our side, so we can do better for the patient down the road. Because it's really, at the end, our goal is to be sure that what we bring is going to be safe to the patient, and then it's going to bring benefits to them. So I think regulatory is one of the even bigger issue than the clinical trial aspect or manufacturing aspect. And if we cannot find ways to improve this area, I think it's going to slow down everything in some way, and that's not what we want. So I am convinced that our legislators are going to understand and are understanding this issue. We know that recently the new PDUFA was voted that allocated more funding to FDA, and particularly more for cell and gene therapy. I had the opportunity to be involved in some of those discussions thanks to the ARM group that is actively working, like many others. But we're going to need more. We need more involvement of FDA, and for them to be more involved, they need more funding and more people.
That's a very interesting point of view. Thank you for sharing. And of course, we cannot help talking about the impact of the pandemic on the pharmaceutical industry. During the past 2 years, the world has learned to operate virtually. How are things going at NKILT? Could you please share that?
Absolutely. Thank you for asking. So NKILT is a small company today. We founded the company a year ago. We stayed stealth as long as we could, because as it's a platform that we are creating ourselves, it's not coming from academia. So we spent the first 6 to 8 months literally building our various patents, writing them. And now we have opened our lab back in May. And we have started to do the primary studies to validate our various patent claims and our IP, and confirm the proof of concept of our new technology. So we obviously looked very carefully about what was happening following and during the COVID pandemic. And what I think is really important to us is that we believe that it doesn't matter where you are when you do your work. What matters is that you are the right person for the job. So one thing that's going to be very important to us is to always prioritize the person that we are hiring for what they bring and not for where they live. I used to say, I don't care to have the guy that lives close to the office. I want the guy that knows how to do the job. So of course, there are some areas where it's not possible to be remote. People need to be in the lab. That's the reality. You cannot do your experiments remote. So you're gonna have to do things with people in the lab, but you can still try to have, to embrace flexibility to ensure that people can have a much better conditions to work. I'm a believer that when people are feeling good, they work better, they work faster, and they're more efficient overall. Then for people that can be remote, there is no reason for them to be in the office every day. What it requires is something that is very important that I saw a lot when I was doing my consulting, helping small company growing. One of the biggest difficulty is what I used to call is finding the right people for middle management. The middle management people are going to be the one that will be able to, not only they know how to do the job, but they also have to be leaders to manage teams. That's a difficult thing. Not everybody can do that. Some people are very good at doing a job, but they're not good at managing people, and that's okay. Not everybody is fit to do such specific job. And in other cases, you have people that are very good at managing teams, but they may not have the skills for a specific job. So we have to figure out where is the best slot. So for us, what we have learned is first, the quest for talent is something that we all face difficulties with. It's difficult to find talent. And when people have talent, they're chased by others. And so it's difficult to retain them. So we need to be really on the listening side, try to understand what would make a difference to make people satisfied by their job. And I think it starts with, of course, the conditions in which you are, the environment, but I think also the philosophy that you bring as a company. We have a purpose. We are trying to bring therapies to help patients with cancer. This is a big purpose to me. And I want to hope that all the people working with me and with our team have the same feeling about it. But we all have to believe that what we're doing makes sense. We all have to believe that we are in an environment that is safe, where people are respected for where they are. And one last point that is extremely important to me. For me, difference is an asset. The difference of people is what makes us better. It's because we come with different point of view that we can be better when we put those differences together and see how we find common ground. So maybe it's a little bit idealistic, but probably because I had the opportunity when I was a kid to grow in multiple places in the world. So the concept of diversity for me is kind of a no-brainer. It's part of my life. But I also believe that it's going to be critical for us to build a company where people feel empowered, respected, and safe. And that will be really the baseline. Whether you work in New York, in California, in Texas, or in Europe, it doesn't matter. You should be treated the same way, and you should be feeling the same way, that your contribution matters and what you bring to the table is important. So that's, to me, is a very important self-centric view. But I will also add one point that is not necessarily NKILT related. But when I saw the work that was done with the vaccine for COVID, and when we had companies that used to fight against each other, competing against each other to try to bring something and be first before the other. And yes, there was still that competitive aspect, but the fact that we were capable as an industry to put our minds together, our thinking together, and bring new vaccines with technology that was not that new. mRNA is known for many years. We just never found the right way of using it. And being able to do it in a record time by doing the right thing all together, I think that was a very good sign that gives me hope for the future, that when we have a serious cause, the science can come together and the people building it can join forces, and that's important to me also.
I completely agree with you on every point, and thank you for sharing this information and your thoughts. I think it's very progressive. And on a closing note, I want you to summarize your own or the role of your company in the field of cell and gene therapy. And where do you see NKILT Therapeutics in the next 3 to 5 years?
Yeah, thank you for that. So first, a few words about what NKILT is about, because I didn't really describe, as I said, it's a new technology. So our approach is really based on, we spent a lot of time looking at the science that was existing, and we were very intrigued about a specific target that we were fascinated about, not only because we think it has some unique properties, but also because it's really truly understudied in cancer. And we believe with my co-founders that it has actually a very important role, particularly in some immunosuppressive aspects that the tumors are generating, whether directly or in the tumor microenvironment. So our target that we decided to focus on is called HLA-G. And HLA-G, if you know about it, you will be very familiar to that, but maybe you don't know. It's what we used to call the pregnancy molecule. It is what actually the fetus is using to protect itself from the immune system of the mother. And HLA-G has the capability of neutralizing all components of the immune system through its natural receptor ILT2 and ILT4. And what we decided to do is that we thought that it would be really interesting to leverage this specific property and turn it against HLA-G by creating a construct, a cell construct, that will target the natural receptor, ILT, instead of using a classic antibody. There are multiple interests in doing this. The first interest is that it's natural receptors, so the affinity will be necessarily the best one because it's the one that nature provided for generation and generation. The second aspect is that it will be fully humanized and will limit any risk of immunogenicity. And the third aspect is that it's a different approach, and it will allow us to, in some way, not only neutralize the inhibitory effect that ILT has on the immune cells, but through this gate, we will be able to actually induce cell killing through our proprietary construct that we are building and engineering in our cells. So we created this technology that we call KIR and not CAR, because we call it chimeric ILT receptor. The concept is the same as what you would do with the CAR, but instead of using an antibody, we use the ILT receptor binding. And we will expand that first in leukemia, and then also in solid tumor, because we believe that HLA-G has a strong effect on the tumor microenvironment, and that if we can neutralize HLA-G in cancer cells, we should be able to have some impact on the tumor microenvironment and hopefully solve some of the issues that we face with solid tumor, for example, that are very heterogeneous and has very strong immunosuppressive effect from the tumor microenvironment. So with that, we decided to move forward, and we are currently testing our various KIR constructs. Our first approach is to go with NK cells because we want to go allogenic, and we want to be able to make also cell therapy accessible to all patients by having a process that will be easier and create an off-the-shelf approach like many of our peers. But the concept is not only for NK cells. We actually also developed KIR T cells, and we could be developing other forms of immune cells in the near future. The interest of HLA-G is also that it's a target that is expressed in over half of cancer. So you have a pretty strong expression in cancers like in leukemia, it's over 50%. In non-small cell, it's between 30 to 70%, depending on the subtypes. In RCC, it goes up to 90+% expression. So there's a variety of tumors that are highly expressing HLA-G that makes it a vast domain for us to act on and to potentially target. Now, switching gears and saying, where do we see ourselves in 3 to 5 years? Obviously, we are very early stage. We are in pre-seed fundraising mode. We need to find more money, obviously, to generate data. We are planning on doing a large seed raising of 10 million end of next year. So from now till end of next year, we need to fundraise again and again. And we do that every day in and out. And if anybody that is listening is willing to look into what we're doing, please join us, and we're happy to discuss. But in 3 to 5 years, hopefully we'll be through our IND process. We will be able to have our first asset potentially going through the IND process, and maybe close to be reaching first in human in the clinic. Like I said, we have a first proof of concept that we plan on doing in leukemia, particularly AML, but we will rapidly also develop our NK cells in solid tumor, where they have yet been able to prove anything in solid tumor, but I know many of our peers are advancing on that field, and by then I'm sure some of them will have shown successful outcomes. So we will learn also from their work. So for me, with humility, I really believe that we hope to be one of the players that will be bringing a new approach in cell therapy that will be complementary to what others are doing. And by doing such, being a contributor to bring better options for cancer patients and starting to put our new options in the clinic. Like I said, yeah, 3 to 5 years, hopefully we'll be close to the clinic or starting the clinic.
So that would be all for today. I want to thank you very much for coming and sharing your thoughts and insights. And of course, we want to wish you good luck with your current research and with fundraising. So good luck.
Thank you. Thank you for having me and for having the opportunity to share my experience and some of my points of view. And of course, if anybody wants to reach out, happy to discuss, debate. We might have different points of view, but I think we're all pushing in the same direction, that is trying to help patients with cancer. And that's what I think is important. Thank you for having me, and have a great rest of the day.
Thank you, you too.
Thank you for watching. If you would like to be our next guest speaker, reach out. You can find the right email in the comment section below. And don't forget to subscribe.